So now we're going to go over the citric acid cycle in some detail and see what all the components are. So we start with acetyl-CoA that we get from the formation of acetyl-CoA, or pyruvate oxidation. Remember, we lost one of our carbon atoms as carbon dioxide when we formed acetyl-CoA. So acetyl-CoA introduces the two carbons that are left from pyruvate to oxaloacetate. Hi, oxaloacetate. How you doing? I see that you have these blue carbons on you. So coenzyme A introduces those two carbons from pyruvate in to form citrate. And then coenzyme A takes off. It decomplexes from acetyl-CoA, but we'll see it again in just a minute here. So the carbons from what's left of pyruvate, this acetyl group here, are now attached to oxaloacetate to form citrate. Now, citrate is the name of the ion. Citric acid would be this same molecule if it had protons associated with all of these carboxyl groups. So that's why we call it the citric acid cycle. So oxaloacetate forms citrate with the addition of these other carbons. Water gets removed and added on in another part of the molecule to form an isomer of citrate called isocitrate. Isocitrate can lose some electrons and one of the carbons to form alpha-ketoglutarate. And you can see that that carbon that's lost comes from the carbon molecule, the carbon skeleton that was already in the cycle. One of these blue carbons here. And these electrons getting added to NAD+, the electron bus, so they can go to electron camp. And we end up with alpha-ketoglutarate. In the next step, we lose another molecule of carbon dioxide. So the last blue molecule of carbon dioxide. So that accounts for all of the carbon dioxide that you're breathing out right now. Comes from your food. And it all comes out either through pyruvate oxidation or from the Krebs cycle. Coenzyme A comes back. Remember, it was up here, now it's down here. And it's going to complex with what's left of our alpha-ketoglutarate. It lost a carbon, it lost some electrons, put them on the electron bus, NAD+, sent them to electron camp. So we've got succinyl-CoA here, which forms at the end, which used to be where oxaloacetate used to be. Okay, so coenzyme A comes in and it leaves again. That's kind of its deal. It's only there for one or two steps, and then it comes in for one or two steps. So it's gone again. And we're going to end up with succinate. And in the process of forming succinate from succinyl-CoA, we're going to be able to produce that molecule of ATP through substrate level phosphorylation. And you can see here we start with GDP, which is guanosine diphosphate, and we can form GTP, which is guanosine triphosphate. Now this is another nucleotide. So it's basically just like adenine. Adenosine triphosphate is a nucleotide like adenine. GTP is just like ATP. And we can easily convert GTP into ATP. ATP is just a more useful currency of energy in the cell. So we get that molecule of ATP. We form that molecule of succinate. And you look at succinate and you can see we don't have anything left but black carbons. We don't know which carbons were introduced at the top of the cycle and which ones are already in the molecule. And why is that? Because succinate is symmetric. This half up here is the same as this half down here. So we don't know which carbons are which. This molecule could be flipped over and we wouldn't know the difference. Now, we can take some more electrons off of succinate to form fumarate. But these electrons don't have quite as much free energy. So they have to go onto a different electron shuttle. They go onto FAD to form FADH2. Fumarate, we add water to fumarate and we end up with malate. But once we form malate, we have a couple of high energy electrons left that we can harvest from malate. Put them on NAD+, the electron bus, form NADH, send those electrons to electron camp, and we have reformed oxaloacetate. Oxaloacetate begins the cycle all over again. So what have we done here? We've harvested a bunch of electrons through NADH and FADH2. We've gotten rid of carbon, all of this very highly oxidized carbon and carbon dioxide, and we formed a molecule of ATP. So now it's time to take the electron buses to electron camp and find out what's going to happen next. What's going to happen next is called oxidative phosphorylation, and we're going to use chemiosmosis to turn those electrons into ATP. So what have we done so far? We've gone through glycolysis, we've doubled down on our ATP, we've sent what's left of glucose, pyruvate, and the oxidized form of pyruvate into the citric acid cycle, pulled off more electrons, we've oxidized that carbon, taken its electrons away, and now we have to put those electrons to work by sending them to camp. So those electron carriers are going to take those electrons to the electron transport chain, which is really what electron camp is. Oh, you might have gleaned from where I said we're going to put the electrons to work that electron camp isn't a fun camp, it's a work camp. So we're going to put those electrons to work to produce ATP via oxidative phosphorylation. Okay, so let's see how that works. This is taking place in the mitochondrion of eukaryotic cells. Remember, the cristae in the mitochondria are all folded up and convoluted to produce a lot of surface area. And the electron transport chain is made of a bunch of integrins, a bunch of integral protein complexes that are within the membranes of the cristae. So these carriers that in the electron transport chain go back and forth between reduced and oxidized. Reduced and oxidized. What does that mean? Basically, it means they are accepting and passing on electrons. And as the electrons get passed along, they drop in free energy. So they drop in free energy. And all along, we've been sort of saying that oxygen's very greedy for electrons, and it will rip electrons away from just about any other molecule that it can, or any other atom. But what that means is that it's also very good at accepting electrons that are very depleted in free energy. So they are accepting of electrons no matter how depleted they are. So in this diagram, we can see that there are these four multi-protein complexes. They have a lot of iron and sulfur associated with them. Iron and sulfur are very good at changing oxidation states, which means they're good at passing electrons along. So you can see NADH is dropping off electrons that have a lot of free energy relative to oxygen. So they drop those off at multi-protein complex 1. FADH2, which you remember was getting electrons in the citric acid cycle, is dropping off its electrons at multi-protein complex 2. They have a little bit less free energy than the NADH electrons, but they are all going to meet up again in multi-protein complex 3. And they're just going to keep passing the electrons down and sort of draining their potential energy, making them more and more stable. And eventually they're going to end up in multi-protein complex 4. And when those electrons are almost completely spent of of potential energy, oxygen comes in and says, all right, I will accept you, you tired electrons, and I'm going to pick up some protons and we're going to form water. And those electrons in water are going to spend most of their time around that oxygen atom, because we know the nature of water is that it's got those polar covalent bonds. So what is this work that the electrons are doing? What the electrons are doing is they are pumping protons from that innermost part of the mitochondrion, the matrix, to that intermembrane space, that space between the inner and the outer membrane. Okay, so why would they do that? They do that because it's kind of like pumping water uphill. They're generating a gradient which we can exploit to produce ATP. So the protons getting pumped into the intermembrane space are going to try and get back into the matrix. And the only way they can do that is by passing through an enzyme called ATP synthase, which synthesizes ATP. So unlike in substrate level phosphorylation, where we took a phosphate group of one molecule and put it onto ADP, we're just going to take phosphate functional groups and ADP and ram them together to form ATP in that way. So that's what we mean by oxidative phosphorylation rather than substrate level phosphorylation. And this is all run through this proton gradient that we've created by using the power of those electrons. And this is an example of what we call chemiosmosis. So here's a cartoon illustrating how it works. NADH drops off its electrons at multi-protein complex 1. The electrons are put to work pumping protons out of the matrix into the intermembrane space. So we're cranking the pH down, making it much more acidic in this intermembrane space than it is in the matrix. FADH2 comes in, and it's going to drop off its slightly lower potential energy electrons, and they're going to get put to work, again, pumping protons out of the matrix into this intermembrane space. So we go through multi-protein complex 1, 2, 3, 4, and each of these multi-protein complexes is progressively more electronegative. But finally, it's going to come into contact with oxygen. And that's where your oxygen that you're breathing in is going to go. It's coming in to accept electrons. It's going to accept those electrons and that have been working so hard to pump protons into the intermembrane space so that they can drive ATP synthase.